Section 5D on vaccines, chemotherapy, and drug resistance. And the first presenter is Bosco Odhiambo Kimathi.
Good morning, everyone. Good morning. I request those who are still chit-chatting to at least settle down so that we can be able to move forward. Thank you. My name is Bosco Kimathi Odhiambo. I work with the Kentech as a senior vet officer. I'm also a student at the University of Pretoria, pursuing a degree in Masters in Global One Health. So this is part of my academic work. It was a study conducted in the Lambwe Valley to find out whether there is resistance of trypanosome species from cattle populations to demeanors in a saturate. Welcome. Now to the background information, trypanosomiasis, as you all know, is a disease of humans and animals, transmitted mainly by the tsetse flies. Then tsetse flies and trypanosomiasis remain a major challenge in Kenya, despite decades of interventions and on control. The Lambwe Valley rangeland has been a locus of tsetse activities since the beginning of 20th century. And people who are familiar with Lambwe Island, they know the impact of tsetse in that area. It even caused migrations of people, and right now people are just trying to move in after the problem has been contained. Now, concerted efforts by various stakeholders have seen a marked decline in the burden the burden of TNT in Lambwe Valley, and this you can find from the references shared there. And then control of trips over the years has relied on the use of trypanosides, vector control, and on the use of trypanotolerant livestock. These compounds are medium-based, rimidazine-based, and isometamedium-based are the most widely used for the treatment and control of trypanosomiasis in Kenya. Now, resistance by trypanosomes to each of those compounds have been reported by various studies, and some of them are highlighted there. And this resistance is largely attributed to improper usage of these drugs. For instance, in Lambwe Valley, there's this study conducted by Ivy Okello et al. That found out that 54% of farmers in that place self-treat their animals. 50% of them use trypanosides frequently, isometamedium being the most used, followed by demeanazine. And then infections are higher in herds of farmers that self-treat. And this one we can always tell why, because they're not able to tell the dosages, they're not able to know the correct diagnosis and such things. Demeanazine and saturate, the drug under study, has been in use for more than 50 years for the treatment of trips and babesiosis. Resistance to demeanazine and saturate has been recorded by various studies. In Kenya, there is scanty of information on resistance to DA in Kenya, despite its widespread use. Now, this study sought to determine the prevalence of trypanosomiasis in cattle in selected villages in Lambwe Valley, Kenya, and also to establish the presence and level of resistance to trypanosomes to demeanazine and saturate in order to inform policy. Now, the study area, as I've been explaining, is Lambwe Valley, which is in southwest Kenya, and now with the current delimitation, it is in Homa Bay County. And this study, we conducted it in 3 villages that are close to each other. That is Kigoto, Wiga, and Gendo. And it was a cross-sectional study conducted in July this year, after the end of the long rains. Now, sampling, we sought to sample 400 cattle, and this sample size was determined by according to the formula described by Cannon and Rowe in 1982, and it was based on an expected prevalence of 5% And the reference for that is the study that conducted by Ivy Okello in 2022. And also, we referred on data that we got from Kentech repository. And as you can see in from 2020, the prevalence has been between 4 and 6%. So we predicted that even for this study, it will be around 5%. Now with that, we've got a sample size of 400 cattle. Now to determine the prevalence of trypanosomiasis in cattle, we collected blood sample through the ear vein in all the animals, in the 400 animals that we targeted. We collected blood samples from the ear vein in the first step because of challenges of personnel. I was the only one doing it with an assistant. So after we collected samples, we processed them through the BCT technique. We centrifuged, we transferred the contents on a microscope, and then we observed for motile trypanosomes. Now, all animals that were sampled, we gave them unique numbers, and then we determined PCVs using the PCV reader and recorded the results. Now, prevalence of trypanosomiasis was expressed as a percentage of the total number of infections, positives, against the number of cattle screened. Now, to determine resistance, now at this stage, animals that tested positive in the previous stage on BCT, we traced them back, and then we collected more blood samples from them. We collected about 4 mils in EDTA tube from the coccygeal or the jugular vein. And then this blood sample was mixed with the cryopreservatives. ESG buffer was used in this case in the ratio 1:1, and 2 mils of that mixture was transferred into cryovials and suspended in liquid nitrogen for 24 hours before they were in mass, so that to reduce a shock to the parasites. Now we preferred block treatment for this study because of challenges of molecular determination of resistance, challenges of it is laborious when you use mice, and then when you also want to do in a wide area, it's also cost expensive, and then it also takes a lot of time. So we decided to go with block treatment, and here what we did. The animals that tested positive on day 1 of the study were recruited into our system. Now their weights were taken with the livestock weighing band, and then they were treated on our day 0 now. It's now the day we started the treatment. It was treated with diminazine saturate at 3.5 milligram per kilogram body weight. Now diminazine saturate, which is presented in 2.36 gram packets, was reconstituted with 12.5 mils of water for injection to give a 7% solution of 15 mils. And then it was administered at 5 mils per 100 kilogram body weight. The vets here, I think they can be able to work out that and determine it was okay. These animals were monitored on day 7 and day 28 for infection status using the BCT technique and also using PCR, and we use ITS1 primers. And this was done at the ECP lab. Now, this is the results for day 1. We were able to collect samples from 395 animals. And then out of these, 19 tested positive for trypanosomiasis. Maybe you scroll. I don't know, there's something. This one is obscuring some things, some details. Maybe you take it up. Okay, thank you. We were able to sample from 395 animals. 19 of them tested positive for trypanosomes, majority of which were T. vivax at 14 and T. congolense at 5. We didn't have T. brucei. And then now the prevalence that we had was around 4.54%. And now the prevalence for TC and TV now, depending on, now this is based on the number of positives, it's 26 for TC and 73.6% for TV. It's not responding. Next slide, please. It's not. Previous flavors are there. Okay. Now, this table here, I know it's busy, but now it gives the overall result following treatment with deimidazine acetate and monitoring on day 7 and day 28. Now, the results of this are summarized here. On day 0, you were able to get 14 positives on BCT and 15 on PCR. There are 2 samples that were positive on BCT but turned negative on PCR. And maybe it could be because of maybe the technician did not identify correctly, or we could have handled the samples improperly. And then number 3, on day 0, 1 mixed infection of BCT showed TB only. There was 1 mixed infection on day 0, that was TB and TC, but on PCR it was TB only. And then on the number 4 observation on day 0 was that there was 1 TB on BCT, but mixed infections on PCR. So PCR it was mixed, but on BCT it was TB. And then the average PCV for day 1 was 19.03%. This is before treatment. Upon treatment, day 7, all the samples tested negative both on BCT and PCR, and the average PCV rose to 23.39%. On day 28, there was 1 death that happened after we left, and it affected even the outcome because some farmers who were neighbors of that animal that died, they did not bring in their animals because they attributed the death maybe to our procedure, which is okay. Though we tried to explain it to them because that animal that died, it was indicated somewhere, but it's number 4115. You realize it had a mixed infection and with a very low PCV of 16. But on day 7, it was negative for trypanosomiasis. So it must have died of other reasons. But nevertheless, you were able to get 12 animals, and of those animals, there were 3 positives on BCT and 4 positives on PCR. And the average PCV for this day was 26.8%. Now, this table summarizes the reinfections or relapses or persistence as we're able to determine upon completion of the study. Now, on day 0, 4102, 4111, 4116, 4118, and on day 28, they showed infections. But now, if you look at 4111, on day 0 it was TB, day 28 it is TC. 4116 on day 0 it is TC, day 28 is TB, 4118 TB, TC. But now 4102 had a mixed infection on day 0. But on day 28, it is TB. On day 7, it was negative. Yeah, so it's an interesting one there. Now, these are PCV trends for day 7 and day 0. And then the uppermost curve is for day 7. The lowermost curve is for day 0. This is PCV trends for the 3 days. The uppermost is the orange one, is for day 28. The middle one is for day 7. And the lowermost is for day 0. This is a graphical representation of the mean PCV across the days. On day 0, you can see it was around 19. Day 7, the mean PCV was around 23. And day 28, it was around 26. This is a gel image for day 7. There's an image missing, but this is a gel image for day 7. As you can see, there was no positive case that was encountered there. This is a gel image for day 0. You can see the positives, the TCs, and the TV against the numbers. Those are numbers of the ear tags of the animals that we recruited here. And then for day 28, you can see here 4102. Oh, this thing is messing me up. You can see 4102, 4111, and 4118 had some infections, and 4116 on day 28. We did subtyping of the T. congolense that we observed, and majority of them were T. congolense savanna, and 2 was a mix of T. congolense kilifi and T. congolense savanna. We didn't have T. Congo forest. Finally, on the main study, we conclude that trypanosomiasis remains a significant problem in the Lambwe Valley, with all the study villages recording some cases. T. vivax is the most prevalent species in the area, followed by congolense. There was no trypanosome encountered. We didn't get any T. brucei. Low PCV, or anemia for that matter, is a cardinal clinical finding in animals with trypanosomiasis, though there are also some studies relating low PCV, saying it's also dependent on the hydration status of the animals. But now from this study, we were able to confirm that anemia is a cardinal sign of trypanosomiasis. And then detection of resistance to deimidazine saturate remains a challenge, and block treatment makes it easier to identify treatment failures upon use through the BCT and PCR. Now, this study did not demonstrate the presence of resistance to deimidazine saturate in Lambwe Valley, and this is evidenced by 100% efficacy on day 7 upon treatment. DA is an effective drug for treatment of animal trips, but it is not ideal for areas with high SSH challenge or where there exists no proper vector control programs. Because we realized by day 28 we had mostly new infections, and looking at the pre-patent period of trypanosomiasis, most studies indicate between 5 to 42 days. Yeah, so there was enough time for development of parasitemia. Yeah, so in areas where there's no proper vector control, DA might be problematic. And then the last one, which is an old-time study, that DA has 0 prophylactic properties. This is very important. Now, on recommendations, number 1, in future, people who want to do block treatment, you can consider inclusion of synthetic pyrethroids in the treatment regime, so that you reduce chances of getting new infections. And then for that sample 4102 that had T. vivax, we are planning to do further tests, but now this time experimental block treatment in goat, because T. vivax doesn't infect mice, and then also the problems with the genetic markers for resistance of T. vivax. So we'll do experimental treatment of the goat to determine whether it was a new infection or a relapse of the first one witnessed. Number 3, I recommend that capacity building for research, especially in public institutions. And this capacity building to include lab infrastructure and technical staff. This is based on my experience on getting one lab that will help me with this work. and number two, on getting technicians who could accurately diagnose trypanosomiasis on BCT. You realize that in the system, most of these people are exiting, they're retiring. So I'm talking from experience, we are going to have problems if something is not done. So sponsors and donors in this, maybe if you see a request for capacity building for research in trypanosomiasis, kindly be gentle on them. These are pictures from the field. This is animal 4105 that had T. congolense. The middle picture is of animals that were brought on day 7. And the last picture is our lab technician. He's retiring next year. And if you see, so the results was very good, because out of 17, 15 were positive. Yeah, so he did a very good job. I acknowledge and I thank these stakeholders for their support in the study. Thank you very much.
Yeah, thank you, Bosco. I now call upon Mochabo Kennedy to come and give his presentation. And in case you left your specs in the ladies, I am not the one who retrieved them. Please come and see me.
Okay. Yes, good morning. My name is Kennedy Mioro Mochabo, working for Egerton University. I'm going to present a project or a study we did in collaboration with Japan, Cairo, and Egerton University. It's entitled Ascofuranon, an antibiotic, ascofuranon, AF in brief, I'll be using AF most of the time, is an antibiotic, is a promising trypanocidal drug. I think the presentation is being put up. Okay. So the collaborators from Japan, maybe they are online, maybe I can mention it by name. The first author is Suganuma, myself, the second author, Mochabo Kennedy Mioro, then a collaborator from Cairo, Chemoliti, Judith, my former supervisors in Japan. I was in Japan for some time. That's Professor Kawazu, Professor Inoue Noboru, and Kita, Professor Kita. Those are collaborators from Japan. I think I mentioned that one. So the ascofuranon is a promising trypanosomal drug for Nagana. Next slide, or do I? Maybe next slide. I don't know, this thing is not working. I try. The arrows across. Okay. Okay, yeah, it's working now. So the outline of the presentation is I'll do a brief introduction, which I don't need to dwell because most of you are experts in TRIPS and SESE. And then briefly, materials and methods, a summary of the experiment, and results and discussion and conclusion. So, as you know, which experts in T and T. I not on that. But there are challenges with the CSA control, and drugs have remained the most practical means of controlling the infections in livestock in many areas. No new veterinary drugs for treatment of AAT that have been released since 1985. And there's an increasing resistance to these drugs. So the drug AF, ascofuranone, there's a chemical structure there, shown on the figure. It's a fungal secondary metabolite of various filamentous fungi, including Acromonium aegypticum, that exhibit diverse physiological activities, including antivirus, anti-tumor, anti-inflammatory, hypolipidemic activities. So there is an urgent need to discover or reinvent or discover new drugs. But this is a study was on efficacy studies of AF. So materials and methods, but for me, it's a summary of the experiment. We had two parallel experiments. We grouped them into T Vivax group and T Congolese group. And we used 3 animals per group. 2 animals served as treatment group, and then 1 animal served as a control per group. So you can see on the figure, on the slide here, experiment 1, which is on top, and then experiment 2, I think, I don't know, yeah, experiment 2, which is T Congolese. So these are the experiments which ran parallel, and the AF, we reconstituted it we used AF around 20 to 30 milligrams per kilogram body weight, but we were reconstituted fresh every day to treat for 7 consecutive days with the glycerol and twin 20. So, and we injected at 2 points, because it's a heavy mixture. You'll see a photo later, a mixture. We were injecting 2 points, 10 ml, 10 ml. And then for 7 days, consecutive days. Then at the end, we finished the first experiment finished at day 43. And then the G Congolese group finished at day 46. But we expanded these trypanosomes in mice before we inoculated into the calves. The colleague who was here before mentioned that T-Vivax doesn't grow in mice, but the strain which we were using was growing in mice. I don't know, I think it was isolated from Nigeria, and it got its way to Ketri as an isolate. And they are listed up there, what we were using, the isolates which are in the trypanosome bank in Ketri, or formerly Ketri, although it's now Biotechnology Research Institute of Cairo. So after 30 days, after the end of the experiment, we took blood from all the calves, and then we inoculated into naive mice to see whether the treatment was properly or if it was effective. So these are the results. The first group, that's T-Vivax group. These are the results. The parastemia of T-Vivax infection trends over AF treatment. Day 6th, we treated day 6th. Sorry. It should go back. Oh, it has gone. Yeah. We treated on day 6 and ended on day 12. So you can see the parastemia trends. It dropped from around day 7 up to day 8. It was 0. And but the control group, we were using BBS as a placebo. The parastemia is high, but on day 7 on the last day of treatment of AF, we administered diminazine acetate as a control with the 7%, at least the normal dose of diminazine. And then all of them didn't have parastemia for the observed period for T vivax. And then you will see later on the trends for T congolese, which had mixed results. So at the end of the experiment, there was no relapse of parastemia. So we were only observing three parameters. That's parastemia, PCV trends, and body weight. Body weight, we are doing it weekly. So this is still AF group, in the T-Vivax group, but these are the PCV trends. The first graph there, and then the body weight trends for the period we observed around for 3 days. And the PCV in all curves showed recovery trend for T-Vivax, and body weight of all curves showed recovery trend. And then the next slide. The next slide now, this is the results from T Congolese group of parastemia. You can see now it's not as beautiful as previous one of T Vivax. You see there is a treatment was from day 9 and ended on day 15. So during the treatment group, the two curves, you can see the parastemia dropping at some point. Then there is a drop. There is a rise of parastemia here. Then somewhere along the last day of treatment with AF, we treated the control group with a diminazine acetate. You can see the parastemia went down, and then all through no relapse. Then somewhere along the parastemia of a treatment group dropped a bit. And then at some point, around day 21, 22, it kind of, there was no parastemia for that period we observed. Up to day 36 there was a relapse of parastemia. So which means the drug did not completely cure the calves. So it relapsed, and then we reach a point. We'll see later on in the PCV trends. We still left them to see what will happen, but the PCV went below normal, and they reached critical point of below 20, and then we intervened with the diminazine acetate to treat the treatment animals. And then that's when we administered at day 49. Okay, I'm almost through. So we treated, and then there was no parastemia after we intervened with diminazine acetate. Then I'm told I'm having 2 minutes, so I'll go very fast. These are the trends of the T Congolese group. You can see the I'm supposed to go back. The PCV trends. PCV in all curves showed recovery trend. No, this is I was talking about this. PCV went to critical below 20. We intervened, and then with diminazine, and then you can see the body weights. But it showed the trend, recovery trend, after intervening with diminazine acetate. So the discussion part of it, T-Vivax group, AF proteasomes were not detected from peripheral blood by parasitological methods using wet smear and HCT. HCT is like PCT, hematocrit centrifugation technique. PCV showed recovery trends and body weight also recovery trends. Trypanosomes were not observed in blood inoculated in mice after observing in mice for 30 days. And the AAT caused by T. virus can be treated by AF 20 to 30 milligrams of kilogram body weight. But then, when you look at T. congolense, AAT caused by T. congolense cannot be completely treated by AF 20 to 30 milligrams per kilogram body weight. Now concluding, AF is a promising tripartite drug, especially in AAT, caused by T. vivax, but not for T. congolense. It suggests that the compound can be developed further to be a sedative drug for T. vivax in non-SESE infested like South Americas and Southeast Asia. In addition, we can further test, that's a further proposal that we and check if it's also as well. And then this is, I don't want to, but this was a scoring way. We are using this chart to score either wet smear or HCT. And then the same thing when you are converting the parastemia. And then these are the photos that you can see the picture there after fresh reconstituting becomes like, looks like penicillin, but it's the compound after we mix with the glycerol and twin 20. And the mice we were using is on the photo there. We were using to make sure that the animals were completely recovered of parastemia. And then acknowledgements, like I said before, this is a collaboration between Kenya and Japan. Cairo, that's where we did the experiment from. I was collaborating from Egerton University. And then the joint funding was from Obihiro University of Agriculture and Veterinary. And this is an institute where I was based when I was doing for my PhD. And then a collaborator also from Nagasaki University. Thank you for listening, and that's all.
Okay, thank you, Kennedy. Now we have Nzumbu Boko talking about vaccination.
Bonjour à tous.
Hello, I'm going to do my presentation in French. I'm called Boko from Central African Republic. I do my research in the Pasteur Institute of Bangui in collaboration with some international researchers. I have the honor to present to you the preliminary results. We're working on the protection in the area of TRIPS. It's experimental still. Thank you. We can observe that whether it's in literature and since presentation made since Monday that there have been good progress in the area of treatment and vaccine production. There have been work and all that is modeling and new technologies. But all this progress, all these efforts have to be renewed so as to face new challenges which are coming. We know all these challenges for resistance. Pathogenic agents adapt themselves to molecules so as to survive. We have also discovered reservoirs on domestic animals or wild animals. We have the vector competence, which means that some flies can adapt themselves and therefore promote transmission. Also climate change, which is also not very well known as for the impact. Now, the elimination and eradication is based on detection, treatment, prevention, and diagnosis. In the case of TRIPS, it is also there is vector control, but also vaccines also can support. Here, I would like just to remind you of the scientific literature which has consensus putting into place of vaccines in the area of parasitology is very complicated. Here I've tried to show you the vaccines. If you see 95% virus vaccines, there are some bacterial vaccines, and also there's also the malaria vaccine which is coming, but we're not very sure about it. There are some teams who are working in silico so as to show the protecting determinants so as to put into place proteins which may be used as a vaccine candidate. Yesterday we had a presentation on DNA model which can also be used. In the case of trypanosomiasis, it is complicated. There are problems of it's very difficult here to put into place an appropriate vaccine. In terms of methodology, we have tried to use a procedure which exists and which has been effective, the issue of nanoparticles. Like, for example, the vaccine for flu, which has shown some effectiveness. So we try to transpose this approach and see what it can give in the area of trypanosomiasis. So with the experimental approach, the parasite was purified and then it was put in the nanoparticle and then injected on mice. Our factor here is the rate of survival so as to assess the efficacy of this combination. There is an injection of the antigen in day 0. 3 weeks later, there's a boost. After 2 months, the parasite is injected, and then the evaluation is the rate of survival. In an experimental model for Gambiense, we have several conditions, but one of the conditions protects at 50% in terms of survival rate, if we see the curve in red. We also model for trip oncolysis, where the same condition which protected at 50% for trips Gambiense protects at 70%. So this is a very early and preliminary results. We shall continue with investigations, optimize the experimental approach, characterize the antigen, because it was a cocktail of antigens which we used, so as to arrive at the characterization of immunity. I thank the presentation, but I think the Minister of Health, represented here by Dr. Duzima, the Pasteur Institute of Bangui, University of Bordeaux, Vaccinano in Lille, in France, cooperation service of the embassy, and the Pasteur Network. Thank you.
Presentation. Our next one is online, 3.24. By Sasea Anosa Soha. If you're online, can you share your slides? Okay. Now I'm looking at 3.26 GE flow. Okay, Flo, share your slides.
Please, can I give this?
Yes, we can hear you. Okay, can you go ahead?
We are fully aware that African trypanosomiasis is a devastating disease, with the animal formerly caused by trypanosoma tuberculosis eradicating a significant landmass in sub-Saharan Africa. So we tilted our focus on this study on some of the biochemical and physiological changes that occur during the life cycle of the parasite and as it undergoes antigenic variation, This is actually the unusual upregulation of a gene called the membrane bound acid oxidase in the bloodstream form of the parasite, and more especially in this substantial amount of inorganic phosphate, which helps in the colonization of the host. The gene is actually involved in the endocytic activity, membranous recycling, and other parasites, and it has been proven to be very essential by RNA interference. So we exploited the political features of the gene to section the gene into different portions, which we concentrated on the extracellular portion without the signal peptides in this work. And it is made up of about 1,293 pieces. So the roadmap of this country uses a transient peptide modified mammalian expression vector to generate our vaccine construct, ascertain the expression of this construct and subcellular localization by fluorescent assay, and then everything out through the localization trial to the construct and give some parameters like the IgG and some cytokines after carrying out the blood infiltration test. Then after which, just the parasitemia and so survival briefing was the tip of our vaccine candidate. Now this shows an overview of how our amplified gene of interest from the native went through the process of subcloning, digestion, ligation, and transformation to obtain our vaccine candidate. And this was confirmed by following PCR here, and then sequencing. And we can see here the sequencing result revealed a change of the amino acids between at position 96 to proline on all the first sequence construct as compared to the reference source from tri-CDE. Here we have a snapshot representation of our obtained construct, showing our antigen distinct line between the restriction enzymes that were used and the treptide and transgene that were used to modify our mammalian expression vector. Now, the IFA result carried out ascertained that the construct would be taken up by the mammalian cells and that the antigen would be expressed as secreted and seen from these screens. We also carried out immunization studies at the earlier stage, and it was two independent immunization trials where we used carefully bred vaccine mice from the same progeny. So the scheme was made up of 4 cohorts, where we have the first cohort, where we gave our vaccine candidates, then infected with 10,000 parasites. We have 3 controls, where we have a positive, where only the vaccine, the P-vax, our construct, empty construct without the antigen was given, after which infection was carried out by injecting the animals with T-Congo, 10,000 parts. Then we have the 2 negative control, which were given just PPS, and 1 was infected at the end. The immunization schedule was made up of a prime and 2 boost gene, and each time before priming we collected blood for assessment of the parameters you can see in red. And then from the after second boost, we collected the parameters again before infecting the animals with the parasites, after which daily parasitemia and survival was monitored. The PI results that it has significantly reduced as compared to the geniazine acetate positive control and the negative control. in Northwood here. The effect, the serum of the immunized mice revealed a significant increase in vitality 10 compared to all the control and the baseline data, as you can see from this figure here. And interestingly, the level of interferon gamma was not changed in all the cohorts of mice, either baseline or control. So from the results of this cytokine, we could see that the vaccine led to an upregulation of interleukin 10 and no effect of interferon gamma, thus enhancing a type 2 cytokine environment in this. Then the next parameter we check, which was the total hemoglobin, showed the value in the serum of the mice significantly increased as compared to other methods. Now, as we check the daily parasitical, we could clearly see that the infected mice show a relative, the main parasitemia of the infected mice show relative. It was characterized by intermittent clearing of the parasite in undetectable level microscopically as compared to all control. Now, the survival rate, the percentage survival rate that was evaluated showed that the animals survived, at least 11% of the animals survived 11 to 35 days after all the
Ça, c'est dans le pour la collaboration. Ça, c'est pas vous qui va aller là-bas. Oui. Tu as déjà pour le coup.
Okay. So, in conclusion, we can see that the candidate elicited both cellular and humoral responses in vaccine mice, and that our vaccine candidate had the ability to switch from a mixed type 1 and 2 environment to a type 2 environment, and by so doing, boosted the resistance of the animal to T-Congo infection. And overall increase of the lifespan of 45.45% was recorded, with a complete survival of at least 60% of mice. Now, the perspective we're talking about is, in future, we are looking at trying to overexpress and purify this process, use it in the real-world trials using adjuvants, and then try to evaluate T cells and other cells in the serum of the animal, while also looking at the stage early and late cytokine production. Thank you very much for listening. I want to acknowledge the following: my university, Africa Center of Excellence and World Bank for sponsoring this work, Bremen for racial donating the modified VVAT, and LECAN, where part of this work we carry out, and the Association of African University, which give them an award of innovation in this for this work in Gambia last year November. Thank you very much.
Thank you very much for the presentation and also persisting through the virtual challenges. I now call upon Carl John, if you're here. Okay.
Okay. Good afternoon, everyone. My name is Guy Callon. I'm head of parasitology at the University of Antwerp in Belgium. My lab is also a drug screening reference lab for DNDi, and we're also part of the lead optimization team in the visualized drug discovery program, which means that actually all the VL leads and clinical candidates have been evaluated in our animal models. So my presentation of today will deal with African trypanosomes, obviously, and a new therapeutic candidates that we have discovered in collaboration with the University of Ghent. So the University of Ghent is specialized in purine nucleoside chemical synthesis, and together we have explored the possibility of looking for new candidates for animal trypanosomizes for the reason that these trypanosomes are completely dependent on the host to take up these purines. And then they have different transporters, enzymes to interconvert these nucleosides to make DNA and RNA. So the molecules we're working with look actually quite similar as adenosine and inosine. You can see the molecular structure here, where we made some modifications on the ribose and also on the R1 position of the base. So, and as a result of this very fruitful collaboration in 2019, we published in Nature Communications a very nice publication on a compound called 3-deoxytubercidine, which actually has a hydroxyl group lagging here and in this R1 position. This molecule is highly potent. It's orally available. It can be used to treat at 6.25 milligrams per kilogram, and it treats trypanosomiasis across the blood-brain barrier. So it's passing very actively across the blood-brain barrier. So we informed the NDI about these compounds. Of course, the NDI already saw the great successes of fexinidazole or caziborole, so they were not really interested to further develop yet another compound for human African trypanosomizes. So we redirected our activities towards animal trypanosomizes, which we also discussed with GalfMed. And so then GalfMed also informed us that we have to be careful because a lot of compounds that have been declared to be active have a liability that often they are not active against Trypanosoma vivax. So we, of course, then tested this compound against different animal trypanosome species. It was highly active against Evansai, Ecuperdum, indicating it may be very useful for Sura and Dorine, but indeed, it was not active against Trypanosoma vivax. So we had to readjust our screening strategy. And so we did that by actually in vitro screening on T. brucei and ex vivo screening on Trypanosoma vivax. And we considered that if we would find activity against brucei and vivax, we hopefully would be active across all the species that are between of them in the phylogenetic tree. So what we did is we screened over 1,000 compounds of a very highly active series of compounds, and we found only 3 that had a desirable activity profile. 3 analogs, which were not cytotoxic whatsoever against human cells, and they were active against brucei, vivax, congolense, also isometamidine-resistant congolense, Evansai type A, B, and Ecuperdum. All submicromolar stability, because if you administer a compound and it's immediately degraded by liver enzymes, it will not be active. So we did microsomal stability assays with the different analogs. You can see all these analogs here, and you can see the shared feature of this sulfur atom here in this position, in the R1 position. And we have used microsomal fractions from both horse and bovine, showing that these compounds are perfectly stable in the target species. So we progressed to in vivo evaluation. So for this, we adopted different animal models using trypanosomes that we received from the ITM, Busher. And so we infected the mice, and at a parasitemia between 10 to the 6th and 10 to the 7th, we started to treat mice with 50 milligrams per kilogram IP to start with. So what we saw is actually a very nice and very fast, actually within one treatment, you see all parasites are gone from the bloodstream. Both for Trypanosoma vivax, congolense, Ecuperdum, and Evansai, which was really very promising. However, we had one disappointment where we saw for Evansai a relapse with one of the mice that was treated. But actually, we saw exactly the same with the diminazine aceturate group. So I think what we can conclude from this is it looks very promising because there's a very fast cidal activity of these compounds, and we didn't see not a single sign of toxicity in the animals. So when we look at new compounds, we also always want to know a bit more about their mode of action. And so the mode of action is sometimes very interesting. It's not essential for further development, but we want to get more insights into how these compounds are working. We combined the RNA interference technique, which is a whole genome RNA interference library that we used from Isabel Roditi, which is an inducible system, and metabolomics. So we were able to show that selective and induced silencing of adenosine kinase results in a 100 to 1,000-fold drop in activity of the compound, which indicates clearly that adenosine kinase is quite important for the mode of action of this compound. We also have seen in the metabolomics that indeed we find mono and dephosphorylated forms of the drug. So together we can conclude that adenosine kinase is essential in activating the drug for this compound to be active. Very fast, and they die with a complete deprivation of proteins. And we also see very aberrant forms with multiple nuclei and multiple flagellae. So indicating some problems at the level of cytokinase. Now, in the context of drug discovery, there's a new initiative at the level of the EU. So it's an EU cost network, which we call One Health Drugs. I'm a management committee member and also work group leader, where we aim for novel drugs for vector-borne diseases with a minimal impact on the environment. So we organize meetings, workshops, training schools, virtual networking tools, and short-term scientific missions, which are typical in EU cost networks. So the aim for new drugs with a minimal environmental impact actually triggered a number of additional tests that we did with these compounds. And so you can imagine for nucleoside analogs that genotoxicity may be a liability. So we used 3 very well-established tests. Vitotox, which looks at mutagenic potential. The COMET assay, which looks at double-stranded or single-stranded breaks. And the in vitro micronucleus test, which looks at aberrant cell division with the formation of a micronucleus. So using the correct positive controls, you can very clearly see we didn't detect any sign of genotoxicity. In the context of ecotoxicity, because you can imagine if you're going to use a compound for veterinary use, ecotoxicity may be a problem as well. So we had the possibility to look at planktonic crustacean, which is Daphnia, how the compound is acting against this, and as well green algae. And in both cases, we saw dose-response curve, EC50 of 17 for Daphnia, which is very well, actually very well accepted as safe by ecotoxicologists. There is some toxicity against algae, but in an acceptable range, both in an acute and a chronic test. So we conclude here again that this compound has a relatively low ecotoxicity footprint. So to conclude, this compound analog 3 is a very potent candidate for treatment of animal trypanosomiasis, all the different species that we have tested. It's another drug class than is currently available, and it also has another mechanism of action. It's depending on phosphorylation by adenosine kinase, and it has a very good safety profile. If you look at in vivo toxicity, we didn't see that. No genotoxicity and no overt ecotoxicity. Of course, we did all this work not to treat mice, right? So we are looking forward to be able to test this in larger animals, as we have seen maybe from the first presentation. And also we are currently negotiating the out-licensing of this compound series to a company. I will stop here by thanking the people involved and my team at the University of Antwerp.
Thank you very much for that presentation on potential new compounds. Now, finally, I think it's Harriet Otti. the last presentation of this session.
Thank you very much. Thank you for the opportunity to present today. It's a pleasure to be here. My name is Harriet Orty. I'm a veterinary epidemiologist at the University of Glasgow. And I'm presenting this talk today on behalf of a big team of people from University of Glasgow, Edinburgh, the Vector and Vector-Borne Disease Institute in Tanga in Tanzania, and the National Institute for Medical Research in Tanzania and Liverpool School of Tropical Medicine. I'm going to talk today about reasons for trypanoside failure. Okay, hopefully we're sorted. Yes. So we know that livestock keepers are very dependent on trypanosides for both treatment and prophylaxis in affected areas, partly because that's one of the options that they're able to implement themselves. And trypanoside use is also a key part of the progressive control pathway. But we also know that there are issues with practices that particularly farmers use for trypanoside use, as well as wider issues with drug quality and drug resistance. What we don't know that much about is the relative importance of those different factors in treatment failures, or how frequently failure happens when farmers or livestock officers are using trypanosides in the field. Next slide, please. Okay. So this work is part of a larger project called Combat. This is the Combat with 2 A's project, not to be confused with the Combat with 1 A. And this was a wider project looking at treatment failure and drug resistance in livestock trypanosomes using laboratory approaches to look at mechanisms and markers, alongside modelling to look at strategies and collection of field data. What I'm presenting today is the field data part. So this was to identify and to quantify reasons for treatment failure in farmer-led trypanoside use. This was done through a longitudinal observational study. So we recruited 30 animals in each of 21 farms. The farmers were not selected randomly. We really needed farmers to engage, so we actually asked people to volunteer to be in this study. They had to be using trypanosides regularly, and in particular using isomethamidium. And then when farmers who owned any of those 30 cattle in each herd were planning to use a trypanoside, they would call our project team. Our vet would go out immediately and examine the animal, take a blood sample before treatment, and then document the trypanoside administration that was being done. Follow-up visits were then conducted for sampling and examination at 1 week and 4 weeks, and then for isomethamidium treatments also at 8 weeks and 12 weeks to look at prophylaxis. And this was all done in Serengeti district in the north of Tanzania. So you can see the location of the farms here around the Serengeti protected areas. So this is quite a high Tsetse challenge area. There's some information here just to summarize the type of farms that were in our study. As I mentioned, they weren't randomly selected. So in general, these tended to be biased towards quite large herds that were more engaged with veterinary services. So average herd size was around 150. Trypanosomiasis was always one of the biggest health concerns for these farms. They were all using insecticides alongside trypanosides, and they frequently saw wildlife while they were grazing, which in this area is a good proxy for proximity to Tsetse habitat. And they all, or most of them, were reporting that they were finding that trypanosides were not working as effectively as they used to, which we had heard quite frequently in this area, which was one of the triggers for doing this study. So over the length of the study, we documented 295 trypanoside treatments, which gives us a trypanoside administration instance rate of around 0.5 doses per cow per year, which is moderately high, although not as high as in some areas. Isomethamidium was the drug that was most commonly reported, but our selection process for the farmers was biased towards people who used isomethamidium, so that's not too surprising. From a different cross-sectional randomized survey, we know that diminizine is actually the most commonly used drug in this area. And to note that people were also using hemidium, admittedly less frequently, and also it was quite common for farmers to mix drugs and to give mixed trypanosides to cattle. When we looked at the doses that were being administered, around 75% were the appropriate dose, but underdosing was most common for isomethamidium and overdosing more common for hemidium. We looked at 4 measures of whether a treatment was adequately administered or not. So one was dose, the second was the route of administration, the third was whether the injection was administered competently or not, regardless of the route, and the fourth was whether the trypanosides were stored appropriately. 66% of administrations were correct for all of those measures. So that left about a third where at least one of those things was not done correctly, and the incorrect dose was the most common reason for it not to be appropriate. The samples that were collected were all tested by PCR. We ran separate species primers and ITS in parallel to increase the sensitivity. We found Congolense most commonly, followed by Vivax and then Brucei. And this is consistent with what we found in cross-sectional surveys in this area as well in cattle. When we looked at whether farmers were using trypanosides because they thought they were treating an animal that was sick or whether they were using it in a healthy animal for prophylaxis, it was about half and half. What's interesting is when they were treating an animal that they thought was sick, we only found PCR positives in 43% of those animals. So most of those, when they're treating a sick animal, it would be diminizine or hemidium. So that's a lot of animals that are receiving drugs that didn't test positive for trypanosomiasis. We then looked at we analysed by PCR all the follow-up data as well. It becomes quite complicated at this point when you've got PCR results over multiple time points. And we wanted to be transparent about how we were categorising treatment failures and treatment successes. So I don't have time to go. I know you won't be able to read this, and I don't have time to go through it. But if anyone's interested, please do come and ask me about it afterwards. And that enabled us to classify each treatment and prophylaxis as either a success or a failure. So this is the results for treatment. We can see that for isomethamidium, diminizine, and hemidium, in most cases, treatment was successful. There were a small number of failures, and they occurred both with adequate and inadequate administration. What's interesting is that even when the treatment was given inadequately, it wasn't uncommon for it still to be successful. When we look at prophylaxis, however, there were 44% of the prophylactic administrations that did not give successful prophylaxis. for 3 months. And those are made up both, about a third of those failures were due to inadequate administration, but that leaves 2/3 of those failures where the drugs had been given appropriately. So that would lead us to think that they would be due to either resistance or drug quality issues. Seem to have got stuck again. But could you move to the next slide, please? I'll try and summarize without my slides in front of me. Overall, this is quite a complex study with a complex data set, so it's hard to summarize it briefly. And there are lots of caveats and assumptions that I'd be happy to talk to anybody about. But overall, our data shows, might one day show, that quite a large proportion of animals that were treated as sick animals did not test positive for trypanosomes, so we really need to consider the importance of diagnosis in that situation. We have identified treatment failures that are not explained by inadequate treatment, and therefore drug quality or resistance are the most likely issues. We are waiting for data on drug quality, so we will be able to differentiate between those two once we have that data. And we're also have completed an intervention trial for resistance, so we will have that information to be able to differentiate resistance from drug quality. Obviously, both are concerning. It'd be nice to be able to show you my last slide. Okay, could you go one forwards as well? Great, thank you. So just to conclude, so our trypanoside failures are suggestive of resistance and or quality, which we will be able to differentiate, and particularly concerning furosemide. But there are other reasons that explain some of the treatment failures around practices that are being used. Obviously, education and awareness of farmers is really important if we're going to address that. And those issues aren't going to be resolved by new drugs. So we must be looking at these in parallel to new drug development. Otherwise, new options that come online are going to have the same issues that we have at the moment. Diagnosis is a huge challenge, and we really need to be investing and advocating for investment in field diagnostic capacity, just in terms of new tests themselves, but also in the infrastructure around that and animal health systems more generally, because obviously that's one of the issues in many of these areas. Thank you to our team, particularly our field team, who had a really difficult job with this intensive clinical monitoring, but managed to collect some amazing data. And just finally, we held a meeting in February in Tanzania focused on trypanoside use and trypanoside resistance. Some of you were there. It was a really interesting meeting, some very good presentations and discussion. If you're interested, most of the presentations were recorded. If you go to the website that's listed there, you can watch some really good and interesting presentations on that from the participants in that meeting. And there's going to be 2 or 3 papers coming out of that meeting in terms of the discussions that were had and the next steps from that. Thank you very much.
Thank you very much, Harriet, for that interesting presentation. So the papers from this session are now open for discussion. And I think in the interest of time, we'll just take 5 questions. Okay, 1, 2, 3, 4. Okay, let's start there. Yeah.
Merci pour la parole.
Thank you for giving me the floor. I'm called Patrice from the DRC. I was interested by this session on treatments. I have questions which I would like to address to Kennedy, who talked about the new treatment, AF. My concern, and I have some curiosity, he talked about treatments, he talked about the behaviors related to this that treatment, if we consider also the strains with the rats and cattle. He says that on the issue of strains, it was effective. When they put it on the rats, it was a little bit complicated. So at once, I want to ask a question to him and to the other presenters who talked about treatment. Does the space or the race of the animal be an important factor which would merit to be considered? Because I observed others also talked about treatment of cattle. Treatment was given in a general way. Does the issue of race become a factor in the effectiveness of the treatment? Thank you.
Kennedy, when the time comes. Okay.
Okay, the same question I'm asking Kennedy. He showed us the results of in vivo assay of ISCTRC. So I wanted to know whether he did also in vitro. And if he did in vitro, did he also see that T. congolense could not be killed by the new drug compared to T. vivax? And if so, what is the hypothesis that he can put forward why T. vivax is sensitive and then T. congolense is refractory? Thank you.
Let's pass the mic to David. Yeah.
Mine is an observation, several observations for Bosco. One is the title of his presentation is Resistance to Trypanocidal Drugs, but he did not display any resistance. In fact, the drug actually worked very well in his hands. So there's a problem there. But there were other issues that I noticed from his presentation. One is if you're going to talk about resistance, and then you give reviews from others who have described that resistance, and it's not just a question of the farmers not being able to weigh the animals properly. I think it would have been good to discuss that, because I believe there are issues of resistance in Lambwe Valley. But even in terms of his cases, there was a case which died, and it had a PCV of 16%. That is a very low PCV, which is probably due to anemia, and it could have died because there was no supplementation in terms of iron or any other treatment, which is why that particular cow died. So it may not necessarily have died from other conditions. It could just have died from trips because there was no further supplementation for that animal. And then when it comes to the diagnostic tests, which is where I was really curious, I was wondering why PCR was less sensitive than the Buffycoat in their hands. Was it a question of the samples, the way the tests were done, the way the primers were handled? That would have been useful information to find out. And then when you talk about Buffycoat technique, you can use it either for detecting motility, if you're using it in terms of looking at the wet samples, but you can also do smears if you're going to do morphology. So which one did you use? Did you use smears or did you use the Buffycoat technique for motility? In which case, how are you able to distinguish the different species? If it's smears, at least specify that. But even with that, you cannot be very sure because there are always some morphological differences. So there were quite a number of inconsistencies that I found. Oh, and the final one was the fact that you decided that at day 7, since you didn't see any parastemia, that the drug was effective is not necessarily true. From the presentation by Dr. Mochabo, which showed that you can have complete absence of parastemia, but a few days later on you have a resurgence of infection. So your conclusions, I think, were subject to other interpretation.
Okay, thank you, David. Ale?
Hello, I'm from Côte d'Ivoire and France. I have a question. I would like to know if the strain which was used for preparing the antigens which were injected, was it the same strain which was used for the heterolog challenge? Because there's a possible interference.
Okay, let's check those questions. Starting with Kennedy. Kennedy is over there.
Yeah, thank you for the questions. I think I've had about 3 of them. One was on the strains of isolates used in mice. We used the strains which I showed you. One was T. congolense was Erie 1180, but there is a number for Ketri. And the one which we use for T. vivax, because T. vivax doesn't expand in mice, but the strain which we used, surprisingly, it was expanded in mice and was growing in mice. So that one, I hope I've answered the one who was asking about the strains. So the strains which we used were growing in mice. So we didn't. Initially, we were saying we wanted to expand T. vivax in goat, but the strain which we used was pathogenic and it was able to expand in mice. Then the second question was AF in vivo and in vitro. In vitro studies were done by our colleagues in Japan, and they were found to be sensitive to both T. congolense and T. vivax in vitro studies. But now in vivo, when we did in Kenya, we expected to get similar results, that the compound will work against both strains of T. vivax and T. congolense, but then it didn't work well effectively for T. congolense. But again, this compound, we were mixing it with glycerol and TIN20. We had a recipe or a protocol now to prepare that. So there could have been also that way of preparing the mixture to get the active ingredient active and administer, could have been maybe a limitation or leading to not getting the desired results. So going forward, maybe this was on a lower scale, using 2 animals as treatment group and 1 as control is a very few. This is a very low sample, but at least it gave us some insights into where we can go. Then there's another question about parastemia. Okay, we were using 2 parameters, parastemia, PCV, and body weights. Although it was a short period, so the body weights were not changing too much. So we were mostly relying on parastemia. And that's why, again, I think if I got the question clearly, the parastemia, we were using both wet smear and BCT. So when there is a relapse, actually when it was in T. congolense, when there was a time there was no parastemia at all, but then we monitored, I think, for around almost 1 week, then there was a relapse. And then when the parastemia comes, it came slowly, and then it was with vengeance. It came more than even when we inoculated the parasites. Then that's why we went further. After treatment, both in the control and treatment group, we went further and sub-inoculated blood from the calves into naive mice to see whether there will be is another way of diagnosing whether the parastemia was, or the animals were completely. I hope I've answered the questions.
Bosco?
Thank you very much. I'm glad my questions came from my professor in vet school, so it's a familiar voice, so I will answer them confidently. Now, it's a new infection. Number two, the technician used the morphology and the speed of the parasite on the wet mounts to determine the species. And then they were not 100% consistent on PCR, because there are two which he diagnosed as either TV or TC, but turned otherwise on PCR. And then on the sensitivity part of PCR, we had two samples that tested positive on BCT, but were negative on PCR, and then I attributed this, either one, maybe the technician was not accurate here, or two, we were not able, maybe we didn't handle the samples properly. Yeah, but generally there was consistency. Generally there was consistency. And then on day 7, we did repeat the PCR for day 7 with the advice from my supervisors. One of them is Dr. Masika. And the results were consistent. And then we went ahead and did species-specific tests using primers. And there was also consistency. So we were supposed to, initially we had planned to do day 14, but we were constrained by resources. But again, if you look at day 28, the results on day 28, there is only one suspect, that is 4102, that is still, again, under investigation. Thank you.
Okay, thank you. Final question was I think to Boko on vaccination.
Merci.
Thank you so much for the question of Alain, which I can consider as a contribution to reassure him it's the same strain, but we had this discussion in our team to see what could that give if it was a different strain. But as I said, in our perspective, we shall be obliged to use other strains so as to see the results, and I think the interpretation may not be the same. Thank you.
Brings this session to a close. I'd like to, rather than give a summary myself, I'd like to just ask maybe Giuliano from FAO to give very brief thoughts, and then I'll also ask Dr. Oksama from WOA to speak. Giuliano, I know it's an ambush, but I know you're always ready.
Thank you, Daniel. I have followed the presentations with a lot of interest. Maybe I have one thought from the FAO perspective on integration of research and control activities. A recommendation, a suggestion is that this research be conducted as much as possible in close collaboration with the national veterinary services and the authorities that are mandated to develop and implement in the field the control activities. This is something that we are trying to promote through the combat project, where interventions. We call these atlases, but these are information systems that should tell us whether we are going up or we are going down. We have seen the WHO presentation. We know where we are going with sleeping sickness. If we ask ourselves where we are going on animal trypanosomosis, at the moment it is very difficult to give answers. So we need a more comprehensive and robust data management to monitor and to plan our actions. And the second overarching strategic push we are promoting from the FAO on animal trypanosomosis is what we call the progressive control pathway. The key of the PCP, progressive control pathway, is to go step by step, is to avoid setting overambitious goals that may not be achieved and that may cause donor fatigue and challenges in resource mobilization. So we believe that many countries like Kenya, Burkina Faso, and all the countries that are developing national atlases are buying into this concept, evidence-based decision-making and progressive stepwise progress. Sorry, I'm not a scientist, so I'm probably not very good to summarize the scientific session, and thank you, Daniel, for the opportunity.
Thank you very much, Giuliano. Now I ask Sam.
Thank you very much. As I told you on the first day, our intervention from the World Organization for Animal Health is really focused on standard setting. And in addition to that, I would like to let you know that WOHA, previously known as OIE, we have a twinning program for laboratories, parent and recipient laboratories, whereby capacities can be enhanced. And you can apply, and if there's funding, of course it's competitive, if there's funding, then we can have this twinning arrangement that will enable your laboratory to become perhaps a collaborating center. And this is what I was saying yesterday, that there's only one reference center for trypanosomiasis, and it's based in Paris at CIRAD. No problem in that, but we also need to have this kind of capacities locally. So this is one area that I think we need to focus on. Can we enhance our capacities? How do we enhance our laboratories to be able to actually address these issues? That's one area that I think I would perhaps talk about. The other thing is that we have a program on antimicrobial resistance, looking at the prudent use of antimicrobials. And here we have talked about trypanocidal drugs. We've talked about resistance. And I would like to encourage you, because we have been collecting this data and we release them every year. I think this is the 7th cycle. But we have had very little data coming from the trypanocidal drug resistance. I know the information is there. So it's nice to know that this information is there and it can be tapped. I think I have much else to say. But I just want to congratulate the the presenters here. It's long since I sat in a scientific meeting. And I'm really impressed by the good data that's coming out. One thing I noted is that, maybe I didn't notice correctly, is that the human side is actually a little bit further ahead. Now, on the animal side, I think are lacking. Is it a matter of resources? Is it a matter of priorities? And what is it? Now, if we are going to promote trade using the standards, I think we need to invest in as countries in our own research systems. Thank you.
Thank you very much, Sam. So I'll now just ask that I'll hand over to the chair, who will now proceed to the next session. It's been a privilege to be a moderator, and I would like to also thank Draman for your work as a rapporteur. Thank you.
A warm round of applause for the moderator and the rapporteur. I also join everyone in congratulating the speakers for the whole morning and the stimulating discussions that this has had. I also was walking around during the coffee break, and I could see that there was very animated discussions going on. I had problems trying to get people back here. But then we still have to manage time. And as of now, much as we are very much in time, we could begin sliding behind, and we shall try to avoid that. I have a few announcements before we move to the next session. The first one is in terms of our travel arrangements. This is an indication that we are coming closer and closer to the end of the conference. All the participants are to fill the circulated form and indicate your departure time, the date, the flight, and the hotel where you would like to be picked from in order for the Secretariat to organize your transport. So the date, the departure time, the flight, and the hotel where you'll be picked from. That way the Secretariat will organize how you will be taken to your departure location. Number two, as I have indicated in previous announcements, we are expecting nominations from the various regions from the member countries. So far, we have not done badly because we have nominees from Western Africa, from Eastern Africa, from Central Africa, and from Southern Africa. We are only remaining with nominees from Northern Africa. We are expecting to have 2 nominees from Northern Africa. So far, so good. Number 3, it has been a plea from a number of delegates that the only group photograph that was taken was with some of the chosen few, with the deputy president. And people were wondering, what about us? I'm informed that if we can organize ourselves quickly, we could assemble at the entrance of this building immediately after this, have the group photo so that it can go into our memorabilia. Finally, it is the poster session. As I indicated yesterday, there is going to be a poster session which is going to be moderated by Dr. Alain Boulanger, and the rapporteur is Nancy Ngari. I would like to request Alain, the moderator, to come and explain to us what is going to happen from now. But before I do that, again, I want to greatly appreciate the efforts of the moderator and the rapporteur of this session. Please give them another round of applause as they leave. And we are still looking for the lady who may have misplaced her glasses in the washrooms. Dr. Blaj.
Okay, good afternoon, everyone. So we will start now our poster session. So the program being slightly behind, I will explain to you how that will take place. After this introduction, we will go for lunch. That will be combined with poster viewing. So if you can eat a bit quickly, and particularly the person presenting a poster stand by the posters as soon as possible for the delegate to have a look at the posters and get the explanations necessary. So I think we'll be back here around, let's say, 2:15, so that we can announce the winner of the competition. Because we will have winners, we have some awards and all of that. A panel had been constituted of five people to judge and rank the posters, and the 5 best posters will have a prize that has not been disclosed neither to me neither to you. And after that, the 5 posters, the 5 owners of the posters will present here very quickly in kind of a pitch what the poster is about, what are the main results. So that should last maximum 5 minutes per person. Okay. The announcement of the winning posters will be done, it's an award, it's a bit like a Miss World or Grammy Award, so we need a beautiful lady to do that. So Verl will be doing that just before the presentation. Okay, so I guess now...